Welcome back to Life Science Solutions. I'm your host, Chris Adkins, and I'm joined today by Nick Letourneau and Ryan Doxey, both Kymanox employees, as we have an interesting part 2 conversation on hyper-concentrated drug formulation technologies and some of the innovation going on in the space. And this is all in advance of an upcoming panel at Partnership Opportunities in Drug Delivery in Boston in October. Ryan, welcome back.
Thank you.
Nick, welcome to the show.
Happy to be here.
Excellent. So glad to have you. So we had an interesting conversation with Ryan, and we talked about the challenges as we push monoclonal antibody formulations really above this or into this 200 mgs per mil. And we're seeing conversations of even into the 300s and the 400s. And some of the issues that start to happen with aggregation and precipitation. Can you just talk us through a little bit of why does that happen, and how do some of these, innovator companies that are out there, how do their technologies solve the problem?
Yeah, when you're dealing with aqueous formulations and you're getting up to the 150 mg per mil or 200 mg per mil range, just the interactions between the antibodies themselves start to become so significant that they aggregate, potentially precipitate. Like you said, stability is really an issue. Mm-hmm. So you also encounter issues with viscosity when you reach those levels. So the ultra-concentrated technologies are able to overcome this by actually dehydrating those monoclonal antibodies, creating these microparticles that are then resuspended in like a hydrophobic carrier.
Okay, interesting. And so, what's actually... Maybe to take a step back and take it from the perspective of, not particularly injection experienced. I think we all have this inclination that we shouldn't inject, particles from a liquid into someone, even if they're particles of the protein of interest, right? In this case. But why is that? Is it a bioavailability concern? Is it a safety concern? Kind of what's driving that? I mean, I think foreign extrinsic particulate is kind of obvious, right? And that's one thing. But what actually causes the issue here in terms of the interaction in the body?
Yeah, so the immune system essentially will identify those microparticles and potentially start to encapsulate them. And
okay.
Yeah, that can cause inflammation, injection site reactions, and just other sort of adverse events. Mm-hmm. Now, the way that these suspensions overcome that, these particles, as soon as they get into a aqueous environment, readily dissolve.
Okay.
So these are rapidly dissolving particles upon injection. There's no depot effect typically observed with this, meaning some sort of extended release of the compound over time. Yeah.
Okay. And so when you say aqueous versus non-aqueous, that means that the liquid, delivery system, for lack of a better term, the liquid environment that they're in just isn't water-based. Is it some kind of solvent or?
Yeah. So aqueous, we're talking about fully dissolved proteins. Mm-hmm. You're not going to see them. It's a clear liquid. It's going to be typically limited to, like we were saying, 150 to 200 mg per mil of the antibody. Then, you have your excipients to help stabilize it. Now, in the suspension, now we're talking about these microparticles, and it's in a carrier, something like benzyl benzoate or ethyl oleate. Another one could be MCT oils.
Okay.
And so these don't have water in them, and it prevents the dissolution of these microparticles.
Okay. For the other non-chemists that are out there with me, what are MCT oils?
So medium chain triglycerides. So these are hydrophobic oils that are known to be safe in the body. So parenterally injected. Now, one thing that will need to be done as part of this technology and qualifying it is the levels, the amount of the vehicle, the delivery vehicle needs to be qualified for subcutaneous or intramuscular administration.
For these delivery vehicles, are we operating above current demonstrated known safe levels, but we think this is safe?
We know it's safe. They're used at higher levels in other routes of administration.
Okay.
And they're pretty natural for the most part. They're just, long chain fatty acid oils.
Sure.
So nothing that is a true chemical. And going back to the particles, one thought that I have, and I think is a great visual. I don't know if you're familiar with the freeze dried candies that are out there now, right?
Yeah.
And as soon as they hit your tongue, they just dissolve. It's very similar to what Nick was describing in these particles. They're dehydrated, so they're hydrophilic. They love water.
Yeah,
right? That's where they're happy. But you suspend them in these hydrophobic oils or delivery vehicles, and they just kind of relax, and they sit dormant, non-reactive for the most part, just waiting for that water environment. So, once you're basically swapping out the vehicle after it's administered into the body, and those monoclonal antibodies react almost immediately.
Yeah. Because of that environment that they're stored in, do you get better or worse shelf life?
I would say possibly better.
Yeah.
But
as long as you keep moisture out of the equation, which hopefully your primary container is doing, right?
Yeah. Yeah. There's lots of potential benefits here, but you think, Nick mentioned the viscosity increase, in aqueous for the MABs above, 150, 200 mg per mil. These oily vehicles are also fairly viscous, but it's a different viscoelastic behavior. The fluid dynamics of these oils are quite different than over concentrated or super concentrated MAB solution.
Yeah, several of them exhibit non-Newtonian fluid properties.
Okay.
So and have shear thinning. So you'll notice once you actually get the injection going, that injection force drops.
Interesting. So does this then play into, if we think long term, okay, you're doing this as a life cycle play because maybe right now you're either dosing more frequently than you want, or if you actually deliver this much drug in one dosing, you're using, we talked about chair legs, horse legs, auto injectors, or you're having to use an infusion pump system, which isn't great from a quality of life perspective. So, but does that then translate to, hey, we need to be considerate of the non-Newtonian nature of the liquid all the way back to the auto injector design and what the force mechanism, the force profile of the spring, I'm assuming a simple mechanical system, but does it impact it that far back?
Yeah, I think it's critical at any phase of development to be keeping the viscosity of your drug in mind, right? So a lot of these start out in pre-filled syringes. So that's where the testing for shelf life, for example, often starts. That's where the initial filling is. Eventually, those pre-filled syringes may end up in an auto injector. But initially, you're going to want to make sure that these can be manually injected. The viscosity of these does tend to be concentration dependent, though. So as you reach concentrations approaching, 400 mg per mil, it may be pretty reasonable. You go up to 500 mg per mil, 600 mg per mil, then you start seeing almost an exponential increase in the viscosity in some cases. Now, that might be acceptable if you have a device to inject, but it might be uncomfortable or difficult for someone to manually inject.
Sure. Interesting.
So I just want to add on this that, we talk about viscosity and, just to get kind of down in the weeds a little bit and, for our viewers who are watching this and, thinking viscosity and, rotational viscosity with a spindle, it's really the understanding the rheological profile of this material. So the shear thinning effects and not just looking at a single point, on a viscometer, but looking at shear rates, increasing shear rates over time so that you can see, okay, where is that shear effect happening? And, am I getting this true, shear thinning profile that I expect? So both the viscosity curves and the flow curves of your product is really key. And you can map that to concentration, right? And overlay your rheological profiles as you increase concentration to see where your break point is.
If we want to get really,
We do.
complicated here, this is where a good point to start looking at the gauge of the needle, for example, can become really important, right? So even if it's a 27 gauge needle, is it a 27 gauge needle regular wall? Is it a thin wall? Is it a ultra thin wall?
Ultra thin wall,
special thin wall, right?
Yeah.
You'll see all the terms out there, right? So based on that diameter, it can impact exactly how the shear thinning occurs.
Yeah. So you touched on some of this earlier in terms of device compatibility, and especially with these technologies that they're still relatively new, certainly new in their regulatory approval process, right? Very, very beginning. But what's the right path for ensuring long-term device compatibility? Because ultimately most people don't want to, dose themselves at home out of a pre-filled syringe. There's a percentage of the population that does, some data shows, right? But most don't. And so what's the right way to, as you're building out your product development plan and your roadmap, to ensure that you're going to keep long-term device compatibility in mind, and you don't make something that you're not going to be able to sell for a different problem?
I would start by really making sure you understand the design space around the device you're trying to use. So, that could be meeting with the various device manufacturers and trying to understand where are the limits and the injection forces you're capable of handling with that device. What are the injection timeframes? And once you understand that, you can start, like we said, mapping it to the viscosity, mapping it to the drug concentrations that may be feasible. It really depends what your priority is as well. If the priority is delivering a small volume, maybe you're okay with a higher viscosity, high concentration formulation that might need to be, either manually injected or have a very specialized device. Like there are some devices coming out now that can handle, I believe, up to 50 to 80 newtons, which is pretty incredible.
So much,
right?
Yeah.
So these are very powerful devices, and that's not always necessary, but it's something that needs to be considered because there are going to be constraints. You need to look at how fast are you trying to inject this, and you can start to look at that back at the pre-filled syringe stage, right? So you're not going into it blind, but you're going to want to set your targets early.
Yeah.
And the clinical path, right, for analyzing or testing these high concentration formulations of MABs can be done in parallel with the device development. So the earlier that you are looking at the device characteristics and using your TPP, your QTPP to guide your development, you can do a lot of this work in parallel. And you don't have to have the auto-injector in the clinical study. But you do have to have a point at which you introduce it. Yeah. And it's usually, later stages after you demonstrate a proof of concept for these high concentration formulations.
Yeah. Certainly after you've managed to introduce them through an IND.
That's right. That's right.
Yeah. Because that's the challenge that we're seeing first. That's right. So I think it's important to take into account this kind of, cradle to grave, soup to nuts, whatever analogy you want to use, but we need to think about the end at the beginning. That's right. As we're developing these technologies. But maybe circling back to some of that manufacturing concern, right? And so we acknowledge here we have a suspension essentially that we're dealing with. So can you talk a little bit about what are the challenges that come from that in a manufacturing space? So how some of us are familiar with kind of these difficult to inspect products from a visual inspection perspective. The facility I worked in, for the most part, we only visually inspected completely clear, liquid product. And then every now and then we would inspect an adjuvant product that was like a milky white, and it was far, far more difficult to inspect depending on what. So has anyone gotten to that stage, and how do folks normally handle, we have other suspension technologies that are out there, but it's another kind of curveball in the manufacturing process for these technologies, right?
Yeah. So we'll take a step back, so yes, you have a suspension. So, the monoclonal antibody, the innovator, is a solution, as Nick mentioned. So sterile filtration is, the sterilizing step.
Yeah.
You now have a suspension, which requires aseptic manufacturing. So no filter is possible, right? Because you have these microparticles that are going to get, they're going to clog that filter.
Yep.
So it has to be done.
You're going to lose all your product at the filter.
That's right. So, aseptic manufacturing. So that is challenge number one.
Yeah.
Is designing the process such that these technologies can be manufactured in an aseptic environment.
You've pulled the historic monoclonal antibody manufacturing. You have a sterile boundary that's around upstream.
Yep.
Your process is self-indicating, so it's actually, no offense to upstream people, I was an upstream guy. It's not that hard from approving the process. But then all of your drug product is incredibly difficult because it's not self-indicating, and you get into media sims and things like that. How far up are we moving the sterile boundary here? Are we moving it to the other side of formulation? Are we moving it somewhere into downstream?
Yes.
Where do we land?
It's the addition of the antibody to the process, right? So, you have the antibody, and you have, the solvent exchange process. We'll call the processes that we know the microglassification process.
Yep.
The spray drying process. So once that drug, you make these dehydrated particles, they're not able to be filtered from there. So it all has to be done upstream of that. Is where your aseptic.
Yeah. So essentially, it's after your purification process,
Correct,
right?
yeah.
And you've made formulation, which kind of traditional formulation at drug product. Again, while I'm offending entire groups of professionals, but formulation for most of these isn't that complicated.
Right.
A buffer exchange or whatever, and getting it to the right volume and the right concentration. But you've made that very, very difficult now,
Yes,
right?
Yes.
And made it where it has to be performed aseptically.
Yeah.
So
no more thaw and fill.
Yeah.
Which
I mean, to be honest, as much as I talk about how easy these things are, it's already hard enough.
Yeah.
So do we move to a place where we're not able to do everything isolator based, and now we're back to ISO 5 rooms?
I mean, I think you can do it in an isolator, but possibly, yeah. I mean, it depends on the scale.
Yeah. Okay.
And I mean, you got to think the kilograms or liters of solvents required. It's kind of tough, to do it in a small contained, isolator type environment with those big bulky gloves on your hands, right?
Yeah.
So I think the shroud has to be taken back a little bit, and you have to look at it from a different lens.
Mm-hmm.
It might be worth stepping back a little bit to explain the manufacturing process.
Yeah.
All right.
Yeah.
So, you still need to create an aqueous formulation of your MAB prior to either spray drying or doing this liquid extraction to dehydrate it.
Mm-hmm.
Some of these technologies are using a organic solvent mixed with your aqueous solution that contains the MAB.
Okay.
To pull out the water into that organic solvent. So imagine you're spinning, right, this organic solvent as you introduce the MAB,
Mm-hmm.
all right, and these particles are forming. The water's getting pulled out. Now you've got a giant mix of these microparticles that have been dehydrated in a bunch of organic solvent. So, how do you get them out? Filtration.
Yep.
Right.
Yep.
So,
Okay.
right. That can be a difficult step because dead-end filtration inherently has some limitations in terms of scale.
Yeah.
So, you're still handling the material and potentially doing that sterile filtration before it goes into this sort of reactor or
Sure.
dehydration area, right?
Okay.
But then after that step, filtration's out the window.
That's a great point. Yep.
Because to your point, at this point, that last filtration that you're doing is the opposite of most filtration that you think about.
That's
You're
right.
keeping the retention.
Yeah, that's right. Yeah.
Yep.
So is TFF an option in that kind of scenario?
It's being explored.
Okay.
I know that
That's where my
there's
brain
been
goes,
some-
but I was a UFDF guy, so it's a logical step, and we never did dead end. So that's a foreign concept.
Yeah, I mean, I think to get the scales that innovator companies need, I think TFF is kind of mandatory, and we've got to make it work.
Yeah, and maybe they need to partner with one of the innovators in that, right? With a Cytiva or someone like that to make that work. So yeah. So that's interesting. So as we talk about the criticality of getting this process to IND, right? There's the manufacturing hurdle, but from a kind of otherwise preclinical evidence, right? I mean, what are folks building out right now to justify putting these, drugs that are made through these processes into phase 1 or into, as I tell folks in my family, right, into otherwise healthy human beings, right? Because that's our phase 1. They're going to be mid 20s, early 30s guys, more than likely, is the stereotype. So what other data, from a preclinical perspective are we seeing that either companies are generating, or are we thinking that the agency might be pushing back on before these go into clinic for the first time?
Yeah, I mean, we're seeing a pretty typical pathway. I'm seeing the rodent models used first, and looking at essentially injection site reactions, seeing if at that scale there's any sort of immune response. But typically what we've seen is going into mini pigs. That's really the next step. That's a little bit more representative.
Mm-hmm.
And then at that point, what I've seen is they're exploring different carriers in some cases to see is MCT oil the best? Is, ethyl oleate, benzyl benzoate, whatever it might be.
Okay.
What's the best fit there? Because they can have different profiles in terms of their injection compatibility, sub-Q injection.
Yeah, what reactions are they going to cause, right? Localized or otherwise? Yeah,
right.
Yeah.
So from there, though, what I've seen is an interest into going to non-human primate studies to demonstrate favorable immunogenicity profiles. Because as you had brought up earlier, you're injecting these particles, right?
Yeah.
So are you going to get an immune response from the particles, or could it potentially activate an immune response against the drug?
Yeah.
The last thing that you want are anti-drug antibodies forming or any sort of allergy. So that's the pathway that I've seen from some of the partners we've talked to.
Mm-hmm. Yeah. I mean, the big thing, the difference here is, you're injecting, as you mentioned, your antibodies, not a small molecule chemical. So, it's not a direct dose response where, it's 800 milligrams more. So you're going to expect the AUC and Cmax to be, perfectly linear. There are physiological responses that the body could impart with that much antibody. Evaluating that.
I think that's a great point. I mean, after you've got the safety portion, tox portion, really starting to dig into how's PK impacted, right? Mm-hmm. So that way you can start to do some dose finding studies and then get ready to go first in human.
Yeah.
Because the ultimate goal with this is to, reduce the patient burden of once a day, once a week to, once a month, once a quarter, once a year.
Yeah.
Type dosing. But yeah, I mean, the only way to find that out is to actually put it into humans.
Yeah. Yeah. And obviously that doesn't work if, to your point, we cause acute side effects because of the protein load is just so high.
Yeah.
Right.
Yeah, that's right.
But obviously from a, lifestyle management perspective, no one wants to have a drug they have to give themselves every week. Logistically, even if you just decide to take a 2-week vacation and you have a refrigerated drug that right. I mean, the logistics there are terrible.
There's all the payer stuff, and I mean, there's a host of benefit to less frequent dosing. I mean, even less plastic out there.
Correct.
From the auto injector and waste.
Yep.
So, I mean,
and not just waste, like medical waste, right?
Yes.
So with specific disposal pathways. So, as we talked about the preclinical side, how important is the early FDA engagement with these kind of novel, technology platforms? What's the recommendation? Where have you seen folks be successful there?
So I've seen them start with the Emerging Technologies program.
Mm-hmm.
So that is a very early engagement where they can start to present their general strategy in terms of manufacturing, in terms of their clinical or preclinical, clinical pathways, what they envision, and start to get that feedback from the FDA early so they can adjust. And in some cases, it's very straightforward. Everything looks good. Other cases, the FDA's, provided some feedback that is very valuable and has helped kind of put these companies going on the right trajectory.
Yeah, I mean, so that point is the most optimal to present the manufacturing plan, right?
Mm-hmm.
So, getting FDA on board with your aseptic manufacturing process is key, right? And I mean, they're scientists and microbiologists as well, and they're very interested in new technologies that add value, and they'll give you really good advice and kind of bring you along the way. And with that early on, it really opens the door for effective scale up, because you have FDA's expectations kind of laid out early on. So
and ultimately they're nerds just like we are, and they work in the same industry as us, and they want to see the same positive impact that we do, right? Everything else that the news might tell you right now, aside from the point. So, okay, so what excites you guys the most about the future of this? In terms of the high concentrated, ultra concentrated biologic space, right? I mean, what about it, motivates you the most as you're supporting folks who are working through this on whichever side of the equation it is?
I love the idea that these drug products that are just on the verge of being able to fit in one of your standard auto injectors will be able to. And it's tough to see, these doses that are just, 5 mils, right? A little bit outside of that 2.25 mil auto injector range, right?
Yeah.
Being able to double that concentration of the drug and just get into something that's standard, that everyone knows how to use and is comfortable with, and that the regulatory authorities are familiar with. I think that's just going to be a huge benefit in terms of the availability of these medications, the patient quality of life, all the other advantages that would come from trying to either use multiple injectors or on-body delivery system and fusion pumps, all of that.
Mm-hmm.
Yeah, I mean, just the potential to reduce that patient burden. You find a lot of patients having to search for different site injections. The longer that we can put in between injections, the better quality of life they're going to have. The less just aggravation from injections over and over again. I mean, that's huge.
Yeah.
So
yeah. And ultimately, from my perspective, the innovation that we see in on-body delivery systems, in infusion pumps, they're great, and they're enabling therapies that are otherwise just completely not doable. But for the layperson to self administer right now, I still haven't seen anything better than the 1 ml or the 1 to 3 auto injector, right?
Yeah,
and it seems to get the job done. And if we can continue to generate them in the force profile, we're seeing, I don't think there's any debate. We're seeing the largest uptick of them in use right now through GLP-1s, and it seems to be working for folks. So I think if we can stay in that kind of range, we're going to continue to see success.
I mean, folks, yes, when appropriate, on-body wearables are great.
Yeah.
But yeah, the average
They're still
person-
better than a horse leg.
That's right. They are better than horse leg. But the average person, wants to inject and go on with their life. They don't want, this bulky.
They don't want to think
device
about it,
somewhere.
right?
Yeah.
They don't have to worry about catching it while they're folding laundry or whatever it might be, right? So, yeah. Excellent. Well, Nick, Ryan, really appreciate you guys, coming on and kind of continuing this conversation. Appreciate us getting the science and nerd out a little bit there too. So that's always exciting. So we're really looking forward to the panel at Pod here in a little over a month and partnering with some other companies, both on the innovator side for these ultra high concentration, but then also on the pharmaceutical side, both in terms of innovators and generic players. Sure. And with Ryan on the panel as well. So I encourage folks who are listening to check that out. Hopefully we'll see you at Pod. And until next time, this has been Life Science Solutions, and we appreciate you listening.
Thanks, Chris.
Thanks.
Thanks for tuning in to Life Science Solutions, presented by Kymanox and produced by Walk West. For more insights like this, subscribe and follow Life Science Solutions wherever you get your podcast. We'll see you next time as we continue exploring what it takes to bring life science innovation to life, because patients deserve better.